Welcome back everyone. Today I will be covering the anatomical and physiological features of the stomach in much more detail. From last week's video, we know that the stomach is a large segment of the digestive tract that mainly functions as a storage and mixing chamber. Its shape and size vary from person to person and even varies within the same individual depending on the food content. Because this is a complex topic, the lectures will be broken down into several parts, and today we will be focusing on the stomach secretions. Let's begin by briefly discussing the anatomy of the stomach. There are four regions: the cardia, the fundus, the body, and the pyloric region. The esophagus opens into the cardiac region at the gastroesophageal opening, which is surrounded by the lower esophageal sphincter. To the left of the cardia and slightly superior is the fundus. Next comes the body, which is the largest part of the stomach. Finally, the body keeps narrowing to form the pyloric region, which consists of the antrum and the canal. This canal opens through the orifice, which is surrounded by the pyloric sphincter before leading into the duodenum. The pyloric sphincter is a thick ring of smooth muscle that plays an important role in regulating the movement of stomach contents into the duodenum by either constricting, which narrows this orifice, or relaxing, which widens it. Now let's talk about the epithelial cells in the stomach. The mucous lining of the stomach is simple columnar epithelium and forms several invaginations called gastric pits, one of which you can see here in this image. These pits are the openings for the gastric glands that secrete acid and other substances. Now there are four main types of specialized epithelial cells in the stomach. There are mucous cells, parietal cells, chief cells, and endocrine cells. Each of these secrete specific substances either into the stomach lumen or into the bloodstream, and we'll cover them one by one. First, mucous cells, like the name suggests, produce a thick alkaline mucus layer that covers the surface of these epithelial cells. The main function of this mucus layer is to lubricate and protect the stomach wall from the acid and the digestive enzymes, such as pepsin. Next, parietal cells secrete hydrochloric acid, which maintains the low pH of the stomach between 1 and 3. A major function of this acid is to kill any bacteria that makes it into the stomach from the foods and drinks that we consume. However, some pathogens like the Helicobacter pylori bacteria have developed mechanisms to resist degradation by the strong stomach acid. This acidic environment also plays a role in denaturing many proteins and increasing their surface area, as shown in this image, so that the pepsin enzyme has more bonds to work on to break proteins down further. Parietal cells also secrete a special factor called intrinsic factor that binds with the vitamin B12 and makes it more readily absorbed in the ileum of the small intestine. B12 plays a crucial role in DNA synthesis, which is important for red blood cell production. Therefore, it should make sense that if there's a problem with the intrinsic factor, less B12 will be absorbed, ultimately resulting in pernicious anemia or a drop in red blood cells. Next, the chief cell's main function is to secrete pepsinogen, which is packaged into zymogen granules that are released by exocytosis into the lumen of the stomach. Zymogens are inactive precursor molecules that need to be activated before they can begin to do their job. Once pepsinogen enters the acidic stomach environment, it is converted to pepsin by the hydrochloric acid that's released from the parietal cells, as you can see in this figure. Additionally, once some active pepsin molecules have been formed, they can act as a catalyst to make more pepsin from the inactive pepsinogen. After pepsin is formed, it can perform its main function, which is to catalyze the cleavage of covalent bonds in proteins, which breaks them into smaller peptide fragments, as shown in this bottom figure. And an important feature of pepsin is that it works best at a pH of 3 or less, which makes it perfect for operating in the stomach's acidic environment. Also, chief cells are responsible for secreting gastric lipase, which breaks lipids down similar to the lingual lipase that's released in the oral cavity. You can remember that parietal cells secrete HCl because parietal starts with P and HCl has a C. On the other hand, chief cells start with C and secrete pepsinogen, which starts with a P. So hopefully this helps you avoid confusing these two cells. The last type of stomach epithelial cells are the endocrine cells. Unlike the other cells we've talked about that secrete substances into the stomach lumen, these cells produce regulatory hormones that are secreted into the bloodstream. Now, there are several types of endocrine cells, and we'll introduce a few here, but we'll discuss them in more detail in the next lecture. The enterochromaffin-like, or ECL cells, release histamine, which diffuse via paracrine mechanisms and bind to H2 receptors on nearby parietal cells, stimulating acid production. G cells secrete the hormone gastrin into the circulation, which also acts to increase acid production. On the other hand, D cells secrete somatostatin, which is an inhibitory hormone that blocks acid secretion in the stomach. Now let's talk about how hydrochloric acid is produced, since it's such a big component of our gastric juice. Now this diagram may seem complicated at first, but we'll go through each step slowly so it makes intuitive sense. We have a blood vessel on the left, a parietal cell in the middle, and the stomach lumen on the right. Now our end goal is to generate acid or protons in the stomach lumen side. First, carbon dioxide in our blood diffuses into the parietal cell. This CO2 combines with water via the enzyme carbonic anhydrase to form H2CO3 or carbonic acid. In step 3, this carbonic acid readily dissociates into a bicarbonate ion and a hydrogen ion. These bicarbonate ions are then transported back into the blood down their concentration gradient via an antiporter, which exchanges a bicarb for a chloride ion. Now this bicarb is a basic ion, so it will raise the pH of the blood, and this phenomenon is called the alkaline tide, which usually happens after we eat a meal. On the other hand, the proton from the carbonic acid is transported into the luminal side via a hydrogen-potassium ATPase. This essentially means that we're breaking down ATP into ADP to drive protons against their concentration gradient into the stomach lumen. Now this should make sense because our objective is to get the protons into the luminal side. Meanwhile, potassium is being transported into the parietal cell and eventually diffuses into the blood via a potassium channel. Finally, chloride ions from the blood are transported into the parietal cell via that same bicarb chloride antiporter that we talked about previously. These chloride ions then diffuse into the stomach lumen via another chloride transporter. Now this diffusion of chloride is important because it essentially neutralizes the positive charges from the protons that are being pumped into the stomach lumen. Therefore, there is less of an electrical gradient for the protons to be pumped across because, as we know, like charges repel each other while opposite charges attract. So hopefully this whole diagram makes more sense now. We can summarize this information using the rule of 4s. For instance, there are 4 main anatomical regions of the stomach: the cardia, the fundus, the body, and the pylorus. There are 4 main types of stomach epithelial cells: mucus, parietal, chief, and endocrine. There are also 4 main components of the gastric juice: the mucus, which is produced by the mucus cells, the HCl and the intrinsic factor, which are secreted by the parietal cells, and the digestive enzymes, which are secreted by the chief cells. Now that's all I have today, so if you guys enjoyed this video, please leave a like, subscribe, and comment. Check out our previous videos if you haven't already done so, and stay tuned for next week, where we'll dive deeper into the regulation of stomach secretions.